Sir, how are you, sir?
Okay, good morning. Have you went through the review that I shared with you? The review article on bioactive natural products.
Sir, I got
I uploaded
it.
in your Facebook group. Have anyone went through? I asked you to respond some questions, like, right, Shafi?
Yeah, yes, sir. But unfortunately, I will go through. Maybe today I will go through this paper.
Okay, tomorrow I need answers from you. So, Shafi, please share the questions on the Facebook group that I gave you yesterday. Not yesterday, day before yesterday, right?
Yes, sir.
Okay. And those who are interested in joining my talk today on the art of writing science, you may join through the Zoom. And mostly undergrad students they are organizing, but it is I think useful for all of you, those who are struggling for writing science. And it is obviously an art, and why it is an art and how can you become skillful in that art is important, because doing science is a part of the story. Writing and publishing science in another bigger part of the story. So, doing science and publishing science together, lead you to become a researcher or scientist. Anyway, let me go to show you some of the slides. And before I start, do you have any question? Joy Devnath, have you joined the Meherpur project?
No, sir. Sanjay didn't inform me or didn't want me to ask me to go.
Why, Rahat? Uh, you are supposed to be communicated.
Sir, I
Sir,
called. Okay.
I can't make a comment about this.
But
I think
anyway, Joy Devnath, you need to visit and join that work because it is close to your house, and you will get lots of insights how we are doing translational research in using high tech. And you will see if not today, but even next time, you can see the lots of differences in wheat by our treatments. We are using nuclear technology, mutation breeding. We are using biocontrol, probiotic bacteria. We are using nanotechnology. We are using selenium. We are doing selenium treatment and many others. So uh, if you go there, you can see the real scenario how we are handling the biotech research in the field level. And our group is the largest group dealing with the wheat blast, maybe worldwide. And we have a significant progress, and I am expecting exciting outcome of the mitigation of wheat blast by our team.
Yes, sir. It is a very interesting topic, and I am very interested. If I get an opportunity, I'll obviously join, sir.
Okay, thank you. Now, in our last lecture, we showed some of the slides on uh, phytoalexins, but I didn't show this slide how phytoalexins are formed uh, in the plant cell. Phytoalexin are formed phenyl propanoids pathway. One of the way of synthesizing phytoalexins are the noids, that is shikimic acid pathway. Shikimic acid pathway result phenyl propanoids. It is also called phenyl propanoids pathway. And phenyl propanoids, there are different hydroxycinnamic acid, coumarins, hydroxybenzoic acid, and all these are the product of shikimic acid. And you know how shikimic acid is produced from the glycolytic pathway and other pathway. Another pathway is mebalonic acid pathway, especially isoprenoids pathway, carotenoids, terpenoids. These are, terpenoidal compound, and these are phenolic compound. So noids are, these noids are present in the phytoalexin formation process, that is phenyl propanoid pathway, shikimic acid pathway, and mebalonic acid pathway. Shikimic acid pathway result phenyl propanoids. It is also called phenyl propanoid pathway. And these are the examples: hydroxycinnamic acid, coumarins, hydroxybenzoic acid. These are the product that is phytoalexins. Mebalonic acid pathway, which is also called isoprenoids pathway, results carotenoids, terpenoids. And combination of these two pathway also can be possible and can happen in some cases. Shikimic acid and polymelonic acid pathway, flavonoids and anthocyanins are produced by this way. So if you are asked how phytoalexins are biosynthesized or formed in the plant cell, then you need to answer shikimic acid pathway, mebalonic acid pathway, and combination of shikimic and polymelonic acid pathway. That means there are multiple pathways are involved in phytoalexin formation in the plant cell. Shikimic acid pathway produced mostly phenolics compound, phosphoenol pyruvate from glycolysis, plus erythrose 4-phosphate from pentose phosphate pathway forms shikimic acid, which are the precursor to phenylalanine and tyrosine. So phenylalanine lyase or tyrosine ammonia lyase, these two enzymes are active there, and they produce these compounds: cinnamic acids, coumarin, p-coumaric acid, caffeic acid, ferulic acid, and sinapic acid. So their structural similarities are like that. Uh, this is all about the story of phytoalexin. But if you browse in the internet, you can find lots of new stories and new advancement in phytoalexin. Uh, and you need to go through to understand more details about phytoalexin, the literature. And in our earlier lectures, I also talked about coevolutionary arm race. Coevolutionary arm race, maybe a few slides I didn't discuss in detail. For example, trichome toxins and their effects on insects. Tomato or other solanaceous plant, they produce routine chlorogenic acid to tridecanone. They reduce larval growth or kill the larvae. And Solanum bartholti polyphenol phenolase system or E-beta farnesin, they can trap the aphids. Similarly, stylosynthesis species produce alpha pinene and sticky resin, immobilize cattle ticks or like that. So Pyrethenum hysterophorus, pyrethenine produced, which is very toxic, even 0.01 concentration, and the trichomes contain quite high concentration. Pelargonium, hortoram, anacardic acid. Anacardic acid also produced by ginkgo biloba and toxic to two-spotted spider mite. Primula of conica, primine antefident. These are the some examples. There are, thousands of compounds have been discovered in many plants, trichome as well as some other, cells of the plant toward of the herbivores. And as we discussed earlier, they are also overcome by the herbivore through different process mechanism, uh, as they have no choice other than getting energy from the plant. Trichome toxins, here is some structural examples, some examples of trichome toxins. So if you are asked, write the name of some trichome toxins, primine from primula, parthenin from parthenium, or E-beta farnesin from solanum, and pelargonium O pentadecanyl salicylic acid. These are the examples. There are lots of more toxins have been discovered. Here, chicory toxin, feeding deterrents of chicory root, stem, and leaf. These are two compounds, lactopicrin and 8-deoxylactocin. Similarly, feeding deterrents of some snowshoe hare in Alaska, pepper birch and green elder plants. Pepper birch and green elder, very famous in temperate zone. And here scientists discovered ferric acid, pinosylvene methyl ether. That means, incredibly diversity in structure of the compound depending on the, plant species as well as their target herbivore. De novo synthesis of proteinase inhibitor. Mechanism of induction of proteinase inhibitors in plants in response to insect herbivore. Insect herbivore not only ward off or inhibited by the secondary metabolites. Sometimes, proteinase inhibitor, specific enzyme inhibitor, which is involved in protein digestion or like that. For example, Colorado beetle feeding the potato leaf. Within 1 to 2 hours, proteinase inhibitor inducing factor BIF is activated. 24 to 48 hours, inhibitor 1 and inhibitor 2 is biosynthesized. They are not actually characterized well. You see molecular weight is 39,000 and 21,000. That means quite big compound, maybe polymeric substances. Inhibition of beetles, digestion of leaf protein. So de novo synthesis of different enzymes, specific enzyme inhibitors are also used as a chemical weapon by the plants to stop the herbivore. This is very exciting. Cryptic or mysterious or obscure phenomena. Here you see new term, epigematic. Epigematic means the bright colors of this glandular poison frog. Here you can see serve as a warning to predators of its noxious taste. Epigematism, from epu means away, gematic means sign or meaning. A venomous coral snake. That means some animals like snake, insects, and even frog, they use some strong color so that the predator birds, they get the warning signal. What warning signal? Warning signal is that they contain noxious taste. They contain noxious, toxin, so that birds predator avoid to attack them. This is really interesting. For example, coral snake, red milk snake, or glandular poison frog. So cryptic metabolize or fate of plant toxin metabolize or excrete also may avoid ingestion by the selective feeding. That means, some of the uh, birds or predator, they somehow have the ability to metabolite or excrete that toxin. But apogemetic and apogemetic mimic, here you can see two-term apogemetic means this one, and it really contains the toxin, not fake. But there is apogemetic mimic, that means durashap. Durashap, contains a bright color, but it does not contain poison. But cobra contains really poison, and it has special in its neck special warning signal. So in that case, uh, the predator, sometimes apogemetic mimic, sometimes can save them, but this is some sort of fake warning. uh, and anyway, uh, these are the way the animals uh, protect themselves from the predator. So not only plants, but also different animals like frogs, snake, even butterflies. Butterflies have huge, uh, warning colors. We love butterfly due to diverse, bright color in their wing. Basically, those colors are uh, the warning signal uh, to ward of the birds, that is predators, because they deposit different kind of cardiac glycosides or other toxin in the wings when the larvae feed on the plant. They get the toxin and they transfer the toxin in during preparation and in the wings. And wings alert contains high concentration of toxin and alerts the birds not to eat them. And some of the color also, are not toxin, but also attract the partner, either male or female. This is a different story, but obviously they are also ecochemicals. Contrasting structure of active and inactive cannabinoids of the cannabis plant. Here you see this is inactive structure. So slight change in the structure can change the structure inactive, and sometimes the predators, they can change the structure after ingestion of the plant. So detoxification of nicotine by house, for example, housefly. Housefly, you can find everywhere in this earth, even temperate or tropical or subtropical regions. Here, this is the toxic molecule. But here you see through oxidation, uh, uh, this molecule becomes non-toxic. That is called continine in insects. So nicotine can be detoxified. That means this type of mechanism, coevolutionary arm race are present, and here specific enzymes are used by the insect to detoxify it. Resistance of fluoracetate poisoning by kangaroos. Kangaroo, they eat plant, and when they take different uh, uh, poisoning substances, for example, fluoracetate, and they use their glutathione and then make it finally glycine glutamate. These are amino acid and S-carboxy methyl cysteine, and these are all useful compound, not metabolically not at all fluoracetate, and can easily convert the poison into the useful compound. So best coevolutionary response by animal to a plant poison. This is the kangaroo. Uh, uh, it is a bright example. Most mammals are fatally poisoned at 1 milligram per kg of this fluoroacetate. Uh, it blocks respiration being converted to fluorocitrate, which is a competitive inhibitor of aconitase hydratase, which then stops TCA cycle at the citrate stage. So this compound is very, very, toxic for the eukaryotic organisms. But kangaroos, by using their enzyme, they can convert it useful compounds so that their TCA cycle is not affected, because TCA cycle in the mitochondria is the important cycle for generating energy from the carbohydrate. Fluorocitrate prevents the influx and efflux of citrate. Red kangaroo is highly adapted to this toxin, but uh, gray kangaroo cannot tolerate. You see, the evolutionary process, all are not equally, adapted to all the toxins. Rat and man feeding barrier is discovered by Butler, that rats fed on a diet containing sorghum tannins are able to adapt to their adverse effects. In these rats, there is enormously increased synthesis of a series of unique proline-rich protein in the parotid glands. These, salivary proteins have a high affinity to condense tannin and remove them by binding to them at an early stage in the digestion process. So, very interesting process. The parotid glands produce, unique proline-rich protein, and that can bind with the condensed tannin present in the, plant sorghum. So sorghum tannins are removed during, in the mouth parts region, in the gut early region, and not allowed to go to the final digestion process. That means early digestion process, they are metabolized, they are separated. But in case of human, human cannot do. The anti-nutritional effects of dietary tannin in mammals are linked to inhibition of digestive enzyme, formation of relatively less digestible complexes with dietary protein, depressed growth rate, and inhibition of microbial flora. So, tannin-rich condensed tannin-rich materials are problematic for human. Tannin, are present in many plants. They are astringent. Persimmon, gab plant contain lots of tannin, and if you take too much, then it would be very big problem for digestion. And obviously, iron absorption and others are also affected. So this is all about today's talk, and I think you can now ask some question.
Sir, hello, sir.
Yes, Kanis Fatama.
Sir, are trichome toxins specific to any type of organisms or other? Because if they are not specific, then it may be harmful for many helpful organisms.
Yeah, it's a very good question. Trichome toxins, basically, they are generally toxic against the herbivores. So, plant, it is a defense mechanism uh, uh, externally placing the weapon. Trichomes are on the surface of the leaf or stem, like that. If you visit the tomato field, you can easily sense that your hand or leg, you will find colored, green colored substances. That is the burst of trichome. Tomatine and other many toxins are there. So trichome toxins are generally toxic for the herbivores, but some of the herbivores over the evolutionary process, over the period of long time, they are adapted to that toxin, but some are not. This is why finally in any trichome toxin, you can find the sensitivity uh, 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 to that toxin by different organisms are different. For example, mint leaves. Mint leaves, contains a glandular trichome, menthol, and many other toxic terpenoidal compounds are there. And we can take small amount of mint leaves as antimicrobial, and the aroma, in burhani and many other uh, uh, you know uh, way we can eat small amount. But if you want to take too much mint leaves, then you can face serious problem. That means dose is a matter, as well as adaptation by different herbivores are different, because herbivores uh, uh, they have to overcome the barriers, chemical barriers, physical barriers of the plant, and the efficiency of overcoming all those barriers by the uh, herbivores are not same. There are differences. This is why, uh, for example, predators, some predators can fully adapted to that toxin, and they have the mechanism to avoid that toxin and associate with the friend plant, and serve the plant. So there is huge uh, uh, variation in tolerance as well as toxicity from the side of insect from the trichome toxins.
Sir,
Clear?
yes, sir. Thank you, sir. Sir, another question is, not all trichomes are toxic present in plants. Is it, sir?
Uh, Trichomes are trichome toxins, chemicals are basically toxin, but over the period of time, some of them becomes not at all toxin. Some insect, get the trichome toxin, and they have the separation mechanism and put them in their wing to use as a toxin against the predator or like that. And some of the toxins we are getting from the plants, they are good for our health. They work like antibiotic, because we are exposed to various kinds of infecting microorganisms. Some of them are also useful for boosting our immunity. And in many ways, the adapted herbivore utilize the uh, original toxin into their own benefit. This is why you can say all trichome toxins are not toxic to all herbivores. There are some specificity.
Okay, sir. Thank you, sir.
You're welcome. Not only trichome toxins, plant produce uh, 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 thousands of secondary metabolites in their cell, even in the seed, roots, tubers we are eating. They also contain lots of secondary metabolites, and originally those metabolites are synthesized by the plant as toxin, so that we do not eat their tubers, fruits, or other roots. But over the years of evolutionary process, we are adapted to them, and not only we human, other animals and organisms also do the same. And this is why I have shown you previously a toxin 1 when it is inactive, then toxin 2 is produced, and this is a coevolutionary arm race. And it has been discovered that neem plant as a director indica, 2,500 secondary metabolites are, discovered by human. You can imagine how many diversity and kinds of compounds, toxins are produced through the evolutionary arm race by the plant. And it is costly for the plant because plant has to spend energy to make these ammunitions. In our country, if we want to become powerful in defense system, army, navy, or air force, we have to spend money. Similarly, in case of plant, to defend themselves from the attack of other herbivores, they also spend huge energy for making lots of secondary metabolites. Otherwise, their growth and development, they could use those energy and they can grow better. When plants are affected by microorganisms or insects, or they are under stressful situation due to abiotic factors, you can see the growth is compromised. Plant growth is also slow down because they divert their energy for the facing the stresses, biotic or abiotic stresses. And this is a compromise because they need to survive first, then grow. So there is a balance. So if you can remove somehow by using pesticide or fungicide, other ways in your crop, if you keep them free from the stress, biotic or abiotic stresses, you can see plant growth and yield is increased. Any more question? Thank you, Fatima. Nice question.
So, is there any other sign beside color to detect the toxin, like scent or something other else?
Is there any
Other signs that animals use to detect the toxin?
Oh,
Like
yeah. To detect the toxin, uh, every herbivore, they have the experience. For example, we can eat only 15 to 20 species all over the world of the plants. Most of the plants, 500,000, 5 lakh plant species are discovered, but we only uh, uh, eat the products from 20 species or like that. Because over the years of experiences, we have the capacity to detect which one is a poisonous and harmful. Similarly, other organisms, they can also uh, uh, limit, they also limit their, uh, how to say, limit their food choices of the species, limited number of species they use as a food plant. Uh, and it is a very interesting phenomena. This is why the earth is still green. If all plants are eaten by all animals, then it would be terrible. And obviously all the herbivores, they have the sensory system, they have the receptor mechanism to detect the toxic substances. For example, bitter, if you test a small amount, you can sense the bitter. You can sense the sour or like that. And bitterness, sourness, these are the defense mechanism of the plant. uh, and in science, the ecological chemist, they use different technologies to detect a very small, minute quantity of the particular toxin. They use the bioassay using the animal and plant interaction. Then they extract the molecule from the plant by using organic solvents, because organic compounds, all compounds are organic, they are dissolved in organic solvent. It is called like dissolve like. That means by using organic solvent, you can get all the organic compounds like acetone, ethyl acetate, or some other dichloromethane. You can use n-hexane to extract the molecule depending on their polarity. And then you can use a different chromatographic technique, including HPLC, for purification of that compound. And after purification, you can check the mass, molecular weight. There is molecular formula, there is a mass spectrometry. And then if amount is sufficient, then you can check NMR, proton NMR and carbon NMR, nuclear magnetic resonance. There is a machine you can get different kinds of carbon and different kinds of proton, that is hydrogen present in the structure. And based on all those spectroscopic data, an expert chemist can elucidate the structure. And this is really an interesting process. And there are databases also. If you put NMR data or mass data of a compound, immediately you can get the information that if it is known, then immediately you get the structure. And if it is unknown, then database will tell you that this is a new compound. In that case, you have to do lots of spectroscopic data. Even you need to synthesize that molecule and get the all spectroscopic data to see identical to the natural one or not. And then you test the biological activity of both synthetic as well as the natural one. And when you find all are identical, then the structure you elucidated is the final, and it is a new discovery. So these are the ways ecological chemist, organic chemist, they are handling to discover the new bioactive compounds.
Okay, sir. I got my answer.
Any more question?
Sir, one question is, sir, what does mean de novo synthesis? It's a internally synthesis or?
Good question. De novo synthesis. De novo synthesis means uh, uh, it is synthesized when plants are plant sense the presence of particular enemy. Then internally in the tissue, a specific gene is expressed uh, after sensing the after getting the signal that is the presence of enemy. So when gene is expressed, you know, a particular protein is synthesized or enzyme is synthesized, and then enzyme is utilized for making a particular secondary metabolite. Because enzymes are important catalyst for every step of a transformation of any biomolecule in the cell. Enzyme is required. So this is the way, sometimes de novo synthesis is compound was present there, but not toxic. So particular enzyme is synthesized through the expression of particular gene to convert that compound into a toxic one toward of the invader or the herbivore. So de novo synthesis means synthesis internally in the tissue through the expression of gene and production of particular protein or enzyme, and then synthesis of a new molecule, bioactive molecules toward of the herbivore. And phytoalexins are synthesized de novo because when it is needed. Uh, uh, it is no meaning to carry lots of toxins if there is no enemy there. Clear?
Yes, sir, clear. So then, sir, another more question. Sometimes, sir, in bioinformatics, we are also using de novo synthesis. Then, sir, can I link these two things, especially when the sequence is not, the gene function is not distinguished? Then before then we called de novo synthesis.
De novo synthesis through bioinformatics.
Bioinformatics, that means sometimes the gene function is not for example, in NCBI we don't have any sequence. That means we want to submit a sequence, and then my question is, there is no reference before.
Yes. De novo synthesis, no reference before, right? De novo synthesis means no existence of a reference. In case of phytoalexin also like that, no a compound actually in the plant tissue, no existence. De novo synthesis of de novo synthesis process is required when you do a sequencing of a new plant where NCBI gene bank has no reference genome. Yes, it is different than the de novo synthesis of,
Okay, sir.
the phytoalexin. That one used the bioinformatics softwares to predict and synthesize the, sequence, complementary sequence or like that. Any more question? No? Then thank you so much and have a good day.